Sridevi, please.
Okay. Hi everybody. So I'm here to present the topic of my research. So I have been 1.5 years into my research now. So I'll be talking about some of the experiments that I did and a few initial results that I got. So I also work on the New Zealand mangroves, but I am more interested in the physiology perspective. So I study how these species cope with the stressful environments because they thrive in saline conditions naturally. So by overcoming hunger, I mean, so I'm trying to assess how much, if I give higher carbohydrates, would they survive better the drought conditions? That is my aim of the research. And I will be talking about how I did that and what kind of results I have got from that. So why is it necessary to study the stress effects on plants? So a lot of studies have shown that forests are important carbon sinks and plants are very important in climate regulation. And we all know that, as is evident from climate change studies, that there is going to be increased frequency of droughts, which in turn will increase the soil salinization levels. And hence we need to understand the mechanisms that plants employ to cope with these conditions. So with regard to this, there has been numerous research which focuses on a lot of species, understanding how these species cope with different drought conditions, temperature, and all that. So two main hypotheses have been come out of this research. So the first one is carbon starvation hypothesis, which states that when a plant is under drought, it does not have enough water. So the stomata closes and there is not enough carbon, and essentially the plant dies. The other hypothesis is hydraulic failure, wherein plants have xylem to transport water. So when under drought, because there is no water, air enters the cavities, which is called the embolisms, and plants essentially die. Now, what these two hypotheses don't cover is that there is another part called phloem, which is more important in transport of carbohydrates in plants, and that has not been accounted in these two mechanisms. This figure shows better. So stomata are gatekeepers. They occur on the leaf of the plant. They allow the entering of carbon dioxide and the leaving out of water, if you all know the transpiration pull stuff. So how does water move through the xylem? It is definitely against gravity because it moves upwards, because plant needs to move water from the ground to the top of the leaves. How does carbohydrates move? So when the stomata open, carbohydrates enter and essentially photosynthetes form. So what are these photosynthetes? It's sugars and starch, right? So that's what they mean by source cell, that is the leaf, and then it enters the sink organs, thereby the plants grow and promote. So what I am essentially interested in looking at is how these mobile sugars, which is similar to the blood sugar levels in our system, and how whether that has a main effect on these plants that help them cope with drought. So I know when I say drought, everybody asks me, why do you study drought in New Zealand? New Zealand does not face a lot of drought. But I'm trying to simulate conditions. For example, in mangroves, they thrive in saline environments, and when rainfall events occur, when there is a dry status like today, they are exposed to more salt levels. So I'm trying to simulate conditions like that. So my drought means that I'm not gonna deprive them of water and study, because that does not make sense. So what is the relationship between drought and salinity? So there has been a lot of genetic studies focusing on transgenic plants especially. So they assess that drought effects and salinity effects are mostly similar, but there is one small slight difference. They both cause low soil water potential. Low soil water potential means that there is water in the ground, but the plants are not able to access it somehow. So when I impose a drought condition, what happens is that over time I will stop watering them. So what happens is that there is a ever decreasing water potential constantly. But under salt conditions, so I'm giving them salt water, they have enough water, but they cannot use the water because it's under low water potential because of the salt in that. So that's that's what we are trying to tease apart, the two effects. So the stomatal closure that was mentioned in these two hypotheses is more of a short-term response, but this salinity and drought, I am interested in looking at the long-term responses. So plants have employed a number of mechanisms to cope with this. One of the interesting thing is that they increase the number of compatible solutes, by which I mean, so when I give them a saltwater drought or saltwater stress, they employ this, the osmolytes like carbohydrates or proline and compounds like that accumulate in the cell, and thereby they maintain their cell turgor and growth, and that's how they cope. So I'm looking at how sugars act as osmolytes and how plants cope with the different conditions of salinity that I'm going to give them. So the sugars, non-structural carbohydrates, as I said, they represent the mobile carbon pool. So what are they? They are the stored carbon. The carbon enters the system and they can use it for growth. They can use it for NSCs. So I'm not interested in the structural carbon, like I'm interested more in the non-structural. So what they compose of glucose, fructose, sucrose, and starch. So there has been a new perspective that says that plants deliberately store some of the carbon that's given to them to this mobile carbon pool, rather than the excess that is used in growth that's going to go to the carbon pool. So as I said, why am I interested in looking? Because I want to look at how these serve as osmolytes in these species for turgor maintenance and drought survival. And yes, so I'm trying to answer the question of how this helps them survive. So based on from this, I'm trying to assess whether the factors of drought and salinity are additive or not for the measured physiological variables, which I'll be talking about soon, and what causes them to die, whether it's the carbohydrates or whether they are not able to transport the carbohydrates, or what causes them to die. And I studied this in the New Zealand mangroves, of course. And halophytes are an ideal system to answer this question because they naturally thrive in salt environments. So it's not like if I give them 25% salt water, they're gonna die. That's why this system is ideal for my question. And yes, so as I said, so I'm looking at how NSCs cope, right? So what I did was I wanted two groups of plants that have low NSC and high NSC. How did I do that? I manipulated the light conditions. So what I did, I collect propagules, I make them into two groups, equal groups, and I collect them all on the same day. I grew them to a certain height, and that's about 40 days. And at 40 days, I started this treatment. So what I did is, one group of plants received low light, which is like 2% for 42 days. At the same time, another group received high light for 42 days. Then I did the former group, was moved to the high light, and this was moved to the low light. So essentially this resulted in two groups, that is the high and the low. Now I haven't really quantified the NSC, but I'm hoping based on my other results that these two are relatively different. And so that's my experimental design. So it's a cross design. So once I have these two group of plants, right, I subject them to the different drought and salinity. So by low drought, I mean that I'll be watering them with salt water for 3 days. I had 3 levels of salinity. Each of them had 3 drought conditions. And by medium drought, I mean that they will be watered maybe 6 to 7 days. And high drought was that once I reached the initial level of salinity, after that I did not water them for like 12 weeks. So they're essentially water stress. So this will be salt stress, this will be a combination of salt and drought, this will be just drought. And then, so yeah, these are the propagules that I collected from Mangafai Harbor, and this is how they looked like approximately around 40 days. So at this at this stage I started to shade them, the experiment that I explained, where I grow them from low light to highlight and highlight to low light. Now, after that, so what all did I measure? So I have divided what the variables that I measured into carbon, water, and the environmental conditions. So environmental conditions, I had them monitoring from the start till the end of the experiment. And what are the carbon parameters that I assessed? The NSC concentrations and the stomatal conductance, photosynthesis. Biomass is essentially the dry weight of the plant. And I also recorded the leaf number, plant height, leaf area for each of the treatments. And mortality was based on visual assessment, so it's more like a visual assessment thing. So what I mean by stem water potential essentially tells you how much of water that plant had at that point of time in a certain treatment. And hydraulic conductance tells you how much of water can move through the xylem at a certain point of time in the same treatment. And I'll be telling you how I did that. So the growth was non-destructive. It was done just using normal measuring systems. So stem water potential was measured using, so this instrument is called the Scholander pressure bomb. And what we do is, so we cut the plant and then we essentially seal it off. And you have this pressurized gas sent to a chamber where you fit the plant in, and once you start increasing the pressure, you note the pressure where the water comes out through the xylem. So that's the pressure that's needed, and that's essentially the water potential of that plant. That's how this is recorded. And so hydraulic conductivity, how this is done is, the seedling is cut underwater here, and this is the xylem embolism meter. So what this does is, this has got two pressure tanks. One has got a high pressure tank, and this one has got a low pressure. Like you can see that there is water in that reservoir. So that's maintained at the atmospheric pressure, and that comes throughout. So that's the lower pressure. So what we do is, once we fit the stem here, you first run the water from the low pressure reservoir and record the flow rate. You can see the flow rate in this system and record the temperature and everything. And then you flush out the embolisms, because obviously you'll have some air cavities. So you flush out the embolisms using the high pressure tank. There are different knobs to adjust these things. So, and then you do that for 5 minutes and then record the low pressure rate again. You do this as many times until the flow rate does not increase anymore. So that gives you the maximum hydraulic conductivity of that plant. So you know how much of air has gone in, and whether the drought treatment had more cavities or not, and what kind of things they did. So that's the thing. And NSC is also a destructive measurement. So you just, after measuring the water potential, I just put them in ice and then I dried them in the oven, and I'm still quantifying the NSC using hydrolysis. So that part is still missing in my research. So once I have the picture, I would know whether the higher carbon had more effect on the conductivity and water potential and things like that. And so these are the initial ANOVA results. So my first question was whether this factor was additive or not. So for that, I teased apart the different treatments. So this does not show the interaction terms. You can see that photosynthesis and stomatal conductance are additive because the individual factors affect that more, whereas for water potential and hydraulic conductivity, it's not significant at all. But when you see the interaction terms, you can see that water potential and maximum hydraulic conductivity are affected more. So here it's not additive, the factors are not additive for these variables, but it's more additive for these two variables. And I'll just show you one graph where just the water potential after 12 weeks. You can see how it differs. So this plot, you can see the 75% salinity, right? And this is the higher carbon that has got low drought, which means it's salt stressed in the higher carbon group. So this is the higher carbon and that's the lower one. And the lower one, you can see that it has got a lesser water potential, but the higher one is maintaining a higher water potential. So I don't know whether because it's the higher carbon that essentially helps them to maintain the water potential, or is it because of the root length or other factors that affect it. So once I have that, then I would be able to better answer my questions. So thank you for listening, and any questions?
Thank you very much for your presentation. Do we have any questions?
Yeah.
What you expect to find in the when you interact two stresses, whether they add to each other or they will cancel each other?
Yeah, that's the question, right? So for photosynthesis and conductance, it's more additive. Yeah. There is no interaction, so the interactions don't affect those variables, but it affects the other two, the water potential and conductivity. Yeah, that was the point of doing this. Yeah.
Thank you.
Yeah.
Any other questions? Mangrove.
Okay.
All right. Thank you very much for your presentation.
Yeah.